Good morning or afternoon or night, whatever time of day it is you are listening. This is Concept 3 Notes for Unit 2 Cells, and we're going to be talking about the cell cycle and cancer. Okay, so just for some context, how does this happen? How do we have an ultrasound embryo or an ultrasound picture, of an embryo developing in the mother's womb? And then it comes out and you have a baby, and then it grows up to be an adult. How is that happening on a biology level? What is going on? Well, my friends, it all begins with a fertilized egg. And this may not be what you thought you were going to be starting or ending your day with, but here we are. So, I'm not going to get into crazy detail on this because this isn't an anatomy course, but I do feel like you need some context to understand the cell cycle, which we're going to be talking about today. So, here are some sperm, here is an egg. One sperm will fertilize the egg. That fertilized egg, once it's fertilized, once the sperm has entered it and their nuclei have fused, we call it a zygote. That zygote, that fertilized egg, is going to go through cell division over and over again. Okay, so we have a sperm and an egg. These are made, males make sperm during a process called meiosis, and females make eggs during a process called meiosis. We will be talking about meiosis later this year when we talk about genetics. So hold on to this thought. Don't worry about how sperm and egg are made. We're going to pick up around here. So fertilization happens, and then that fertilized egg starts doing mitosis over and over and over again. So this cell division through mitosis is making a ton of identical cells. So at the beginning of the fertilized egg's life, if you will, if you believe that's when life starts, it's just a clump of identical cells. They're all the exact same. Then a key process called differentiation happens. This is a process that creates special structures and functions. So the cells start to differentiate and specialize. So those identical cells will specialize, and then certain cells that have similar functions will group into tissues, and those tissues will organize into organs, and those organs will organize into organ systems. And eventually that makes up the entire organism that is the developing embryo in the womb. And then that embryo becomes a fetus and is born and hopefully grows into healthy infant and toddler and kid and adult. So, again, another picture you may not have expected this morning. This is the female reproductive system. Okay, so ovaries will release an egg once a month, and then that egg can get fertilized by sperm. And the fertilization is happening here in the fallopian tube. So females have 2 ovaries and 2 fallopian tubes that then connect back to their uterus. So the fertilization happens as the fertilized egg is traveling back to the uterus. This is when the cell division is happening. This is when mitosis is happening. So you can see how the cell will divide, and now I have 2 cells, and then those 2 divide, now I have 4. Those divide, I now have 8. By 72 hours, we have this clump of identical cells that we call the morula. And then at 4 days, we call it a blastocyst. But you can start to see how there's going to start to be some differentiation that's going to happen here. Around 6 to 7 days after fertilization is when the fertilized egg will hopefully implant into the lining of the uterus. And then that is where the developing embryo would be hopefully develop into an actual growing baby. If this does not happen, that is when a female cycle happens. So again, we're not going to get into all those details, but I just like to show you this image that as soon as fertilization happens, cell division starts to happen. Within 30 hours, so less than 2 days of fertilization, we already have 8 identical cells, and then those will start to differentiate. So cell differentiation, what's happening there? These undifferentiated cells are called stem cells, and they can become differentiated into one or more types of cells. So you have that group of cells That's all just the same. They're all identical. Those will start to differentiate into neurons and enterocytes and hepatocytes and cardiac cells, osteocytes, all these different cells that will then organize into tissues and then organs like the ones pictured. Stem cells can basically be found in two places. We have embryonic stem cells, which are cells that have never differentiated and can become any type of cell. So these are the cells that are happening in those first several days of a fertilized egg's life, if you will. And then there's also adult stem cells. So each of you listening to this video, you have stem cells, you have undifferentiated cells in your bone marrow. The only difference is they're already partially differentiated. They can only become, they can't become anything. Like your bone marrow cells cannot become neurons in your brain. They can only become certain types of cells, but they're still relatively undifferentiated. So you start out as this clump of identical undifferentiated stem cells. And like I mentioned in concept 1, the cell is our most basic unit of life. So remember from concept 1, cells are made of organelles, and then there's also molecules in there. Remember our macromolecules like carbs, lipids, proteins, nucleic acids. And then from unit 1, we also talked about atoms. But the cell, that's the first thing that's considered life on this scale. None of these things, organelles, molecules, atoms, are considered life. They don't have the characteristics of life like a cell has. But then cells, those cells will start to differentiate and organize into tissues, which are just groups of cells working together for a common function. And those tissues will organize and differentiate into organs, or a set of tissues working together for some sort of common function. Your organ system would be all the organs that are working together for a common function. And then all of those make up you as an organism or an individual member of a species. So an example could be one of your stem cells, one of your undifferentiated cells, goes through the process of differentiation and becomes a muscle cell. That muscle cell will organize into a group of other muscle cells and become a muscle tissue. And then that muscle tissue will be a part of the lining that makes up the stomach. So there will be other tissues that make up the organ of the stomach, but muscle tissue is one of them. And then the stomach is one of the many organisms that's considered a part of the digestive system as a whole. And then you have a ton of body systems, your skeletal system, muscular, digestive, urinary, cardiovascular, etc., that make up you as an organism. So that's how we go from that original picture of you're just one fertilized egg, and then you're a clump of identical cells, and then you become this developing embryo in the womb that then is born as an individual organism made of all these organ systems and organs and tissues and cells. So that's the context for what we're really going to be talking about today, which is the cell cycle. So this is the process that's happening, like I said, fertilized embryo or fertilized egg, right away it starts doing cell division and making identical copies of itself. That is the cell cycle happening. It's a repeated pattern of growth, DNA duplication, and cell division that's happening in eukaryotic cells. Okay, so we're talking plants, animals, fungi, protists. That's what we're talking about here. And its two purposes are growth and repair. So this cycle of mitosis is happening a lot in those first few days of life when you're just a clump of identical cells, but it still happens after differentiation. So think about right now, your hair cells are growing and they're dividing and making more hair cells. That's how the hair on your head or your face or your legs is growing. It's growing because the cell cycle is continually happening. It's happening in your skin cells. It's happening in your blood cells. Blood cells are dividing and making more blood cells. Skin cells are dividing and making more skin cells. Okay, so this is a process that's happening throughout your entire life. And the cell cycle consists of 3 main phases, if you will. There's interphase, which is, you can see from the picture, this is the majority of a cell's life is an interphase. It's usually growing in this phase. Then there's mitosis, which is the actual part where we're starting to divide the cell. And then cytokinesis is when the cytoplasm actually separates and you end up with those 2 cells. And cytokinesis starts at the end of mitosis. So we're going to go through this. Hopefully a lot of these terms are familiar to you. And hopefully we're just building on something that you've already learned in life science. Okay, So as we talk through these phases, I will have pictures that I think are really important for you to sketch in your notes. Now, one thing I want to make note of, if you look at this cell, there's not a lot of organelles in there. That's because when we're drawing phases of the cell cycle, so that the diagram isn't really complicated, we kind of simplify the diagram so that it's only the most essential organelles for cell division. So typically in all the pictures you're going to see today, we'll have the cell membrane, and then we'll have the nucleus with the DNA, and then we're going to have our centrioles, or the centrosome, which is 2 centrioles with the spindle fibers coming out of them. And then, of course, you always have cytoplasm. Okay, so that's all that's going to be pictured. But just know, other organelles are there. The Golgi and the rough ER and ribosomes, those don't disappear by any means. But just for the sake of simplifying the picture for you, so you're not drawing these really complex diagrams on every page, this is what we're going to be drawing to represent the cell cycle. Okay, now that I've prefaced that, let's get into interphase. So this is known as the growth phase of a cell's cycle or of a cell's lifetime. An interphase is divided into 3 parts. So all the different parts here. There's G1, S, and G2. G1 is known as the gap 1 phase. And during this time, the cell is just growing, it's making proteins, it's just living a normal life. Then there is S phase, and this is arguably the most important of all. This is the synthesis phase. This is where a process called DNA replication occurs, and we will learn about DNA replication in Unit 4 genetics. But essentially, it's the process of doubling the number of chromosomes. This is because when the cell divides, we want both of the cells that result to be identical. And if we don't double your DNA, when the cells divide, each cell would only have half of your DNA. It would only have half of your genetic information. And then when those would divide, they would each only have half of the half. So 1/4 of the original amount. And it would just keep going until you'd have cells with virtually no genetic material in them. And remember, your DNA is the instruction manual that makes you who you are. So we have to have that information. So during S phase, synthesize means to make. We're going to make more DNA. We're going to double the DNA that you have so that then when the cells eventually divide at the end of mitosis, each one will have a full instruction manual of your DNA. After the S phase is another growth phase. It's G2 or gap 2 phase. More cell growth is happening and more protein synthesis. So more proteins are being made here. At the end of interphase, so before jumping into mitosis, the cell should have 2 full sets of all of its chromosomes. So we should have twice as much DNA as before. And this is an extremely regulated process. If there's a mistake made, your cells normally catch it before they divide and keep copying that mistake into future cells. So that's really important, and that's something we will talk about a little bit. Now, before we can go any further, I've been saying words like DNA and chromosomes and instruction manual and all of this. We have to pause and hit some vocabulary related to DNA. This is tricky because there's a lot of words referring to a lot of things that are basically the same. But I really want to introduce them now. I want you to start learning them now so that down the road, as we get into our genetics units and our heredity units, these are going to be terms that you really understand. Okay, so we know that DNA is deoxyribonucleic acid. It's one of the 4 macromolecules your body can't live without. It's the instruction manuals for making proteins. A chromosome is a word associated with DNA. A chromosome is just one long continuous thread of DNA, and it has thousands of genes on it and also regulatory information. Now, what is a gene? A gene is a section of DNA with instructions for making a protein. So we could take your DNA and we're going to bunch it up into chromosomes or organize it, that's a better way to say it, into chromosomes. As a human, your body cells, so that's your skin cells, your hair cells, your muscle cells, your bone cells, your brain cells. They have all of your DNA organized into 46 chromosomes. So your DNA, it looks like this. It gets bunched up and condenses into 46 chromosomes. And each of those chromosomes has thousands of genes on them and has the instructions for making thousands of proteins and also regulating all the processes. It's like a think of it as a really long book organized into 46 chapters, if you will. Now, if that wasn't complicated enough, in your body cells, your chromosomes occur in pairs, and we call those homologous chromosomes. So, if we go back to that original slide, you were originally made with one sperm that came from your biological father and one egg that came from your biological mother. Those fused, and that was the first time you as an individual had your entire instruction manual of 46 chromosomes. Because 23 of those chromosomes, half of them, came in your biological father's sperm, and the other 23, the other half, came in your biological mother's egg. And when those fused for the first time during fertilization, that was the first time that you were you with all 46 chromosomes that make you uniquely who you are. So, oftentimes in your cells, those 46 chromosomes organize themselves into 23 pairs of homologous chromosomes. So, chromosome number 1 from dad and chromosome number 1 from mom are near each other, or are a pair, if you will. They don't have to physically be in proximity. And chromosome 17 from your biological father and chromosome 17 from your biological mother will be a pair. So we're going to talk about this more later, but I feel like I have to just at least preface it now. So again, in one summary sentence, your DNA is organized into 46 chromosomes, and which have thousands of genes, and those provide the instructions for making proteins, which run your body and make you who you are. Now, if you were hoping we were done with the DNA vocabulary, you would be hoping wrong. So these are terms that are really essential for understanding the cell cycle. So a chromatid is one half of a duplicated chromosome. So remember, I told you before mitosis, there's the S phase of interphase, and we double our DNA. So a chromatid is just one of these, and once it doubles, we are going to double it. It has an identical copy. That's this other part. That's the sister chromatids. So two identical chromatids are just two identical copies of the exact same chromosome. So when you see the chromosomes pictured as an X, that is going to be after S phase. That's our duplicated chromosomes. Now, the center part is called the centromere, and this is kind of condensed region of the chromosome, and it's where they appear to be pinched, and that'll be an important part as we get into what's going on in the cell cycle. And then another part that's important are the ends of the DNA, the ends of the chromatids or the chromosomes, and these are the telomeres, and these actually shorten each time the cell divides, and we'll talk about those repercussions later on. So one more time, let's talk about what's happening in S phase with these DNA. So before S phase of interphase, you have 46 chromosomes that look just like this. And we would call these unduplicated homologous chromosomes. So you've got this green one maybe from your biological father, this pinkish purple one from your biological mother, okay? So you would have 23 pairs or 46 individuals of these, okay? The S phase will happen of interphase, and that's where DNA replication will occur, and that's where we double them. So see how it's now an X. Now I have sister chromatids for each one, and I now have duplicated homologous chromosomes. So we would still say that there's only 46 total chromosomes, but we would say they're duplicated. So we have 46 duplicated chromosomes. Or if you're going to count the individual chromatids, there would be 92 of them in a human. And this number is different for different species. Fruit flies only have 6. Humans, though, we have 46 in each of our body cells. So drawing this, I think, and labeling this in your notes is really important. Because again, at the end of S phase, we should have 2 copies, 2 full sets of all of our chromosomes. All right, now let's get into the mitosis. Hopefully this is the part that's more familiar to you from 7th grade life science, or whatever grade you took life science in middle school. So this is the actual division phase of the cell cycle. We divide to multiply. So one cell divides into 2 identical daughter cells. So we're going to go from this to this, and they're going to look the exact same. So the general phases are prophase, metaphase, anaphase, telophase, and cytokinesis. Oftentimes I refer to it as PMAT for prophase, metaphase, anaphase, telophase. And sometimes people will also refer to this intermediate step as prometaphase, but just depending on who you're talking to and what textbook you're reading, they'll include prometaphase or not. What's important to really know, it's not about these individual steps as much as you understanding this is a cycle and it's a process. We can't necessarily just take a snapshot and capture the whole phase because it's a continual thing that's happening. It's just like if I was going to take a picture of you every year on your birthday, that's great. And we could say this is your year 1 picture, your year 2, your year 3, but it doesn't capture all the growth and change that's happened throughout that entire year because it's a continual process. So, but for the sake of understanding, we're going to simplify it. Now, also for the sake of understanding, for your pictures to make them simpler, we're only going to show 4 duplicated chromosomes in our picture. Keep track of that number. Don't draw 4 X's in your first picture and then 7 in the next one. You have to do it the same amount every time. So follow the pictures exactly. But note, if this was a human cell, we would have 46 X's in here, okay? Representing 46 duplicated chromosomes. So what goes down in prophase? Your chromosomes, which are typically looking like spaghetti, which we call chromatin, they condense. And they are now visible as sister chromatids or as these X's. The nuclear membrane disappears. It starts to dissolve. That way the centrioles will be able to get at the chromosomes to divide them. Spindle fibers, that's these what look like spider legs, start forming out of the centrioles. All right, metaphase. Think middle, metaphase middle, metaphase middle. The spindle fibers connect to the centromeres, so that central condensed region of the sister chromatids, and they pull the chromosomes so that they line up single file in the middle of the cell. And this is so we can make sure they're all there. Anaphase, think away or apart for anaphase. The sister chromatids separate and they're pulled away from each other. So we now have individual unduplicated chromosomes on either side. And then we start moving them to opposite ends of the cell, or some textbooks will refer to these as the poles of the cells. Now telophase happens. Telophase, y'all, exact opposite of prophase. So everything that happened in prophase, we're going to do the opposite. Now the chromosomes are going to decondense. They'll start looking like chromatin again. So more thread or spaghetti-like rather than rod-like. Your nuclear membranes will start to reform around the chromosomes at either end of the cell. And then the spindle fibers that came out of those centrioles, those are going to start to break down, so we're not going to need them anymore. And then cytokinesis begins. And that is the division of the cytoplasm, so we have 2 completely individual cells. And this looks different in plants and animals. Plants form a cell plate midway through and the divided nuclei, and it gradually forms into a membrane, and then they kind of split. Animal cells, there's a cleavage furrow. It kind of, the sides pinch in until it separates into 2 equal parts. Regardless of the mechanism, the end result is 2 identical body cells. So if you started with 1 heart cell, you got 2 identical heart cells now. You started with 1 hair cell, you got 2 identical hair cells now. So how often is this happening? Well, it depends. Every cell divides at a different rate based on its need. So for example, the internal lining of your intestines, those intestinal cells, they divide every 5 days, pretty frequently. Your skin cells are about every 2 weeks. That's why you need to wash your sheets at least every 2 weeks, if not every week, because you got some dead skin cells that are falling off into your sheets every night when you're sleeping. Red blood cells every 4 months. If you've ever donated blood, you'll notice they won't let you donate blood again for at least 4 months. You have to have time for the donated blood that's been taken to regenerate. Your liver cells only divide once a year, which is why liver damage is especially concerning, because your body doesn't just regenerate liver every 5 seconds. It takes it's only happening every year. Okay, so why do our cells divide? Go back to the beginning. Growth and repair. Cells don't just get bigger to grow either. You're made of trillions of cells, not just like 10 giant cells. This is because we want our cells to stay really tiny so that they can have a really high surface area to volume ratio. What that means is this will allow your cell to be really efficient energy-wise, and it allows, think back to Unit 2, cell transport, things to move in and out of the cell much more easily if it has this high surface area to volume ratio. So that's why they divide and we make more and more and more. Also, think about it this way. Let's say you were just 10 giant cells. If something happened to one of those cells, that's 1/10 of your cells that's now messed up. Whereas now, your body's made of trillions of cells. So if something happens to one of those cells and they mess up, hopefully it's not that big of a deal and it doesn't cause an issue because you have all these other cells. Now, that's not always the case though. So let's briefly talk about regulation. I'm sorry these are so long. I've gotten extremely long-winded, but I just love this topic. How do our cells know when to divide and when not to? And then what happens if the regulation of this process fails? Let's see. So your cell cycle is controlled by a chemical control system. It starts and it stops events in the cell cycle. And proteins are playing such a key role in this. Remember, I told you your chromosomes have thousands of genes. They also have just tons of regulatory information to control things. And this is external and internal. So signals will come from outside of the cell. Maybe a hormone or some nutrients will signal the cell, hey, you need to divide. But a lot of it's just internal. Signals from the cell's own nucleus, its own DNA will say, okay, it's time for it to divide and make another one of me. A key part of this is checkpoints. A checkpoint is a critical point where stop and go signals regulate the cycle. Think about when you were younger and you would play red light, green light. Red light means stop, green light means go. That's what's happening here. There are multiple checkpoints that are marked with the no sign. And oftentimes our signs are in no, okay? Or we're in the stop position, if you will. These are some of the key checkpoints. So during G1, there's a checkpoint where the cell will stop and it will check if it has enough nutrients to go on, if it has enough growth factors to keep growing, if there's no DNA damage before it'll move on. It can also decide to go into G0 or a resting state and not move into S phase quite yet. Okay, there's another checkpoint in S phase that checks for no DNA damage or replication errors. There's one in G2 that's also going to check that there have been no DNA damage and no replication errors. It's going to make sure the cell is the right size to divide. And then there's also a checkpoint in mitosis that's going to make sure that the spindle fibers have attached to chromosomes correctly and everything's lining up well. So this is a highly, highly regulated process. And again, normally the cell's kind of in that off or that stop position, unless there's some sort of stimulus saying, hey, let's divide. So checkpoints play a key role in regulation. Another thing that plays a key role is apoptosis. So I mentioned this term to you in concept 1 when we were talking about lysosomes. But apoptosis is programmed cell death. Basically, internal or external signals will activate genes that tell the cell to make self-destructive enzymes, and it will essentially destroy itself. Now, examples of why this is important and actually a good thing. When you are in the womb, you can look at this picture right here. You can see your fingers and toes are webbed. Because apoptosis happens, that gets rid of that webbing. And there can be a mistake sometimes where it doesn't happen and you're born with some webbing there, like we see in this picture of this foot. If you hate feet, I'm really sorry that I just put this on your screen. But this is an important process. It's a good thing. And it's also good because oftentimes if we notice an error, apoptosis can be triggered to happen and that we can destroy that damaged cell before it makes a copy of itself and we have more and more damaged cells. So it's an important and helpful process. Now, if we don't catch these mistakes at checkpoints and if we don't destroy messed up cells and apoptosis, cancer can be a result. Cancer is uncontrolled cell division. It's when the regulation of the cell cycle breaks down and cancer cells just divide and divide and divide. They divide much more often than healthy cells do. And because of this, it creates tumors. A tumor is just a clump of cells that's dividing uncontrollably. And you can see here how it's starting to build up on itself, and that's where that tumor is forming because this cancer is happening. So you have some sort of normal cell. Something changes that cell's DNA so that then it becomes a cancer cell where its information is wrong and it keeps dividing when it's not supposed to. So those tumors can form, and there's two types of tumors. Benign tumors are abnormal cells, but that remain clustered together. They can be potentially harmless and easily removed. My father has skin cancer, and they're all benign tumors so far, and he just has to regularly get them removed. They're all contained, they haven't spread. But it doesn't mean they're always easy. You can have a benign brain tumor. So here's a picture of a brain tumor. It can be benign in that it's totally clustered there, but it could still cause great harm where it is and be really difficult to remove. Malignant tumors are where cancer cells have broken away from the original tumor and they've moved to other parts of the body and they're starting to create more tumors. And that movement of and spreading of the cancer cells is called metastasis, or the verb is to metastasize, where the disease spreads from one organ to another. So this is much more severe. This is when my mother was diagnosed with breast cancer. It was deemed malignant because she had multiple tumors in multiple parts of her body, not just the original tumors that were in the breast tissue. So what causes this? Well, y'all, there are so many things, and if we knew all the things, I'm sure we could have much greater abilities to prevent and cure cancer. But in general, here are some of the things that cause it. And we're going to go into these more and do some more explorative stations on these topics too, because I know a lot of you find this really interesting and it's really relevant because you know someone with cancer. So there are biological factors, things like age. You may inherit a genetic mutation. For example, BRCA1 is a gene that gives you a genetic predisposition to get breast cancer. I think of my mom. She was in her late 50s when she got breast cancer. It's much more common to get breast cancer at that age than when you're 30. The type of skin you have. Like my dad is 100% Irish. He is a second generation. Like his parents came over from Ireland to America. So I guess he's a first generation American. His skin has such low levels of melanin. It just makes him really susceptible to skin cancer. So those are biological factors. Also, lifestyle choices, what you eat, your physical activity, how much you tan and expose yourself to UV radiation. Those are all things that can affect whether or not you get cancer. Also, viruses and other infections. We've actually found out that there are viruses like HPV, human papillomavirus, that can cause cervical cancer in women. So that's important too. And also kind of good news because we can hopefully prevent. There are vaccinations you can get to prevent HPV, which can help reduce your risk of cervical cancer. And then also just exposure to carcinogens. Carcinogens are cancer-causing agents. They're chemicals that cause cancer by mutating the DNA. These are things like tobacco smoke and asbestos, which is found in a lot of older built homes, etc. So those are just some examples. These are probably the longest notes we'll ever take. I'm so sorry, but hopefully you found them interesting, and I'm excited to talk to you more about them in class.